So good evening to everyone. Hope you could hear me. Thank you so much for joining us for our Beth Jacobs Got Talent series. This series showcases our Beth Jacob members and gives them the opportunity to share their unique experiences and perspectives. And we're so blessed at Beth Jacob to have members, all of you, who are quite accomplished in your respective fields, reflecting the idea that being a committed Jew, a Torah committed Jew, doesn't preclude engagement with society, but rather promotes it. And one member who is an exemplar of a Jew deeply committed to Torah while making an extraordinary contribution to the world is our speaker tonight, and that's Dr. Shlomo Melmed. Dr. Shlomo Melmed is the Dean of the Medical Faculty at Cedar Sinai and Executive Vice President for Academic Affairs. It's not an exaggeration to say that he's a world-renowned endocrinologist who has helped build Cedar Sinai into one of the best hospitals in the country. And if Shlomo is not in shul on Shabbos morning or not at my Chumash year, it's usually because he's with Lonnie at some medical conference around the world, sharing his medical knowledge and his experience on pituitary medicine or endocrine tumors. He's been named one of America's top physicians, and he's received Cedar Sinai's Pioneer in Medicine Award, among many other awards. So we're so proud to have as members and leaders of Beth Jacob, Shlomo and Lani, who is a force in her own right. And this is a very challenging time for our country, for our county, for hospitals, with the surge in COVID cases. And Cedars is fortunate to have Shlomo in his leadership capacity, especially at this time. So we're so thankful to have this opportunity to hear Dr. Shlomo share his perspectives on COVID and medical discovery. So blessed to have Shlomo as a member and leader in the shul. And it's so nice to see all of you, and so thankful that you've joined. Looking forward to hearing Dr. Shlomo Melmed's perspectives and looking forward to his insights. Okay, turning it over to Shlomo.
Thank you, Rabbi. Well, can you hear me?
Yeah, we could hear you fine. Also has raised a wonderful family as well, children and grandchildren.
Thank you. The last time a rabbi spoke so nicely about me was at my bar mitzvah. So it brings me back many decades. Thank you. Can I share my screen now?
You should be able to. You're a co-host. Go for it. If you want, there we go.
Okay, perfect. All righty. So what I'd like to do over the next 30 or 40 minutes, hopefully less, is go over with you some of the recent advances that are taking place at Cedars today, both for COVID and beyond COVID, and then hopefully has a chance for questions and discussion and editorializing. There are a lot of doctors are listening, and they can probably contribute more than me, and I'll be welcome to answer any questions. So, you know, we were all working and trotting along very nicely in January and February of 2020, and suddenly this virus hit us. This virus with these little golf tees stuck into the ball. You can see these little golf tees, the spike proteins, which act as little suction cups and suck onto any cell which they come up against and thereby do all their damage. And really what happened is that within a matter of days, COVID clouded every aspect of our lives with complete uncertainty, because nobody had ever experienced such an illness before in recorded medical history. So really, whatever we're going to talk about now is really being compressed into a matter of 30 or 40 weeks. In the New England Journal of Medicine, one of our very erudite faculty members, Dr. Kittleson, wrote a beautiful piece about the invisible hand of treating COVID at Cedar Sinai. And she concluded her statement with a very poignant comment that during a unique time in history, the consequences of our suppressed panic, and we were panicked, and the anticipatory dread of the future, because we never knew what the future would hold, are really quite impossible to predict. So within a matter of days, we really geared up as the largest healthcare provider on the West Coast to serve our patients, to serve our community, and hopefully to serve society. And one of the first challenges which we faced was that there was no test to diagnose COVID. The laboratory testing, as you may recall, during the February, March, April, even going into May, was very, very inaccurate. And so doctors working in the ICU were seeing patients with this terrible pneumonia and really had no accurate way of making the diagnosis. And our biomedical engineers came up with a very smart four-dimensional approach to diagnosing COVID in the lungs. Four-dimensional because the three dimensions you can see, and the fourth dimension is the color. And the color of yellow or gold, which you can see in these lungs, really is a reflection of COVID. And that is in real time, in a real patient, lying in an ICU, an image of the COVID disease attacking and ultimately destroying lung tissue. The blue is the remnant healthy lung tissue, and the gray are the lung airways which carry the air to the lung tissue. So this was an opportunity for us to provide the world with a new diagnosis for COVID with a very unique pattern which reflected the disease. And we learned very quickly that there were about 15 days of the disease. On this axis, you can see the disease severity going from north to south. And on the x-axis, going 5 days, 10 days, all the way out, giving you the time. And it turned out that in the first 3 to 5 days, there was a normal, expected viral response to any viral early infection with an effect on the white blood cells, the lymphocytes. And then suddenly, after about 5 days, there was this massive infection of the lungs. This is what we called stage 2 with the abnormal chest imaging. And after stage 2, a third stage of the host response. And the elevated inflammatory and heart biomarkers really contributed in a very, very significant fashion to this disorder. And again, this whole spectrum of disease, this classification of disease, this description of a disease was brand new and literally developed within a matter of weeks. So our first question was, what is this virus that's infecting our patients? And we got our molecular pathologists to work very quickly to actually analyze the genes of the viruses that were infecting our patients at Cedars-Sinai. And we were very surprised to find, and this is an article which was published in the Journal of American Medical Association, JAMA, which is one of the most widely read journals in the world. And this we published very quickly to show that what we had all thought that the virus that we were seeing in Los Angeles came from China was wrong. And in fact, the analysis of the genome of the virus infecting patients in Los Angeles and specifically at Cedars-Sinai actually originated in New York. So there was a New York transmission route which caused the infections in Los Angeles in early 2020. And it also turns out that that New York origin was Italy. So the virus which we have in our patients was transmitted Italy, New York, Los Angeles. And in order to tackle the virus, we decided to approach it with 3 factors: to try and develop antiviral treatments, anti-inflammatory treatments against the inflammation in the lung, and to treat the patients during the recovery phase. For the antivirals, I'm going to show you some examples of some of the antivirals which you've all read about in the newspapers, many of whom had their pivotal clinical trials at Cedars-Sinai, and then show you some work on the anti-inflammatory, literally putting out the fire of the lung response to that virus. And finally, what are we doing for patients who are recovered from the disease, but who are by no means free of the disease? So since the beginning of March in 2020 until January 2021 today, we developed no fewer than 100 approved clinical trials for COVID. And this really kept us busy for the entire year. North of the red line are those antiviral drugs, some of which you've read about: remdesivir, the antibody from Lilly, convalescent plasma. And south of the line are the anti-inflammatory drugs to try and block the response of the lung to the virus. And I want to just point out a few of these, and the ones in the box on the right here are ongoing right now as we speak, because as you've read, we have a very, very heavy load of COVID patients right now. Literally, as we speak tonight, there are hundreds of patients in Cedars-Sinai with COVID, and many of them are benefiting from these clinical trials. Remdesivir and the antibody from Lilly, you've all heard about. What you may not have heard about is the BCG vaccine. And some of you may know, for those of you certainly who come from Africa or Asia or Australia, BCG is a vaccine given against tuberculosis. And it turns out that people who've received BCG for tuberculosis are in fact protected from COVID. Not universally, but as a population group, they are protected. So the BCG vaccine was a very early vaccine, which we started at the end of April in giving the individuals who are actually working in the ICUs to try and protect them. And there've been a lot of studies and literature, some which were published this week, showing the benefits of BCG, the anti-tuberculous drug. But these are a whole host, and every patient has a specific clinical criterion which makes them a candidate for one of these trials. Two of the drugs which you've read about, remdesivir and the antibodies, neutralizing antibodies, were both published in the New England Journal of Medicine with Cedars-Sinai as the lead authors. We were the main sites for these trials which were published. And I want to just emphasize that although remdesivir and the antibody are used now under emergency use authorization and then ultimately approved for COVID, they improve the disease, they improve the severity of the disease, but there are no proofs yet that they improve mortality. So we still don't know that the ultimate mortality changes with these two drugs. And these are the only two approaches out of all the ones that I've shown you that so far have achieved some modicum of success in dampening the effects, the negative effects of the disease. While this was going on, we still continued treating patients. And so what our doctors did with a very, very strong push from the clinical community was design guidelines. How do you treat patients who are not infected with COVID, but who still require heart transplant? So we wrote the guidelines for heart transplantation in the era of COVID. How do we treat patients with chemotherapy for cancer while COVID is going on? How do we allow our women to continue to deliver safely in the presence of COVID? And these three very important guidelines emerged from Cedars-Sinai and hopefully have benefited society and COVID patients and non-COVID patients worldwide, because we're all afflicted with the virus, but unfortunately, disease continues. One trial which I want to talk to you about, and some of you may actually be part of this trial if you had antibodies tested at Cedars-Sinai. And that is what is called what we've termed the CORAL study, C-O-R-A-L-E. That's the Corona Risk Association and Longitudinal Evaluation Study. This is a unique study. There are only 8 in the world. We're one of the 8, in which we are doing comprehensive analysis of all our patients with COVID, frontline healthcare workers treating inpatients with COVID, the close contacts of all our patients, the regional contacts of our patients with or without risk of COVID. We're doing electronic health record-based tracking in real time of all risks and outcomes after they go home. We're doing continuous data collection and surveillance of these patients with remote monitoring, web-based monitoring. And ultimately, all of this data will be contributed into a huge meta-analysis database from the community. We're even sending kits for blood to be tested and mailed back to us to be able to analyze. And so far, 3 groups of individuals have emerged who are at high risk, both for COVID and, most importantly, for the remnants of COVID after the patients are discharged. Hispanic patients are particularly prone. African Americans are prone, and those patients with hypertension, obesity, and diabetes. And these 3 groups are really the ones that we're focusing on to try and follow and get a picture and understanding what this disease is all about. Because remember, a year ago this time, most of us had never even heard of COVID. And now here we are a year later doing comprehensive assessment, follow-up, and full blood analysis going forward in a longitudinal way, including antibodies, including DNA, including protein, even including stool bacteria. Comprehensive analysis of our patients. Another example of what we're doing is in the terms of stem cells. Our scientists have developed stem cells of the lung to grow on a chip. We call them lung chips. And these lung chips are perfused in the blue and the red with fluids as if they are arterial and venous blood. And we add the virus to the chip. And then we add different screening drugs to try and find molecules. In this case, it's an antisense nucleic acid to see which one will actually improve oxygen transport, will improve blood flow in the lung. And at the bottom, you can see the photograph of these lung stem cells growing on a chip, which are completely synonymous with the lung cells in a normal human alveolar cell, lung cell. And on the top, you can see the cartoon depiction of the airflow, the blood channels, and the oxygen diffusion. So this is an amazing, it's not science fiction, it's real. We can actually take stem cells from a lung, grow them up, and grow the human lung on a chip and use it to infect COVID and to test for drugs without having to go through the long process of clinical trials. Another use of stem cells for COVID, and this was a real first out of Cedars-Sinai, was the impact of COVID on the heart. Look on the left. So what we did was we took the stem cells from a patient's heart, and this is done for patients who are undergoing cardiac biopsies for other reasons. They sign consent. We derive stem cell from their biopsy tissue. The stem cells are grown in a dish. We grow the heart in a dish from the heart stem cells. We infect them with COVID. And look at the difference in the top, the beating heart. This is a beating human heart in the dish, derived from a patient's stem cells, not infected with COVID. And at the bottom, you can see that the heart cells are beating very, very slowly, if at all, when infected with COVID, which you can see in the red fluorescent staining. And this was the first demonstration that not only is COVID affecting the lungs, but it is also directly affecting the heart muscle, all derived from stem cell work rather than from the patient as a holistic body. But, so those are some examples of what we're doing in terms of COVID research. But of course, life continues after COVID, and all of us get sick. And COVID is only one component. And so our research continues in all the non-COVID areas. And over the next few minutes, I want to give you some examples of the ongoing research right now as we speak. So what we've done is, for those of you who've driven past, have seen the Cedars-Sinai sign in what used to be the Pacific Design Center. And we've really converted the Pacific Design Center into a huge innovation laboratory. And this is just an example, a photograph of one of the floors in the Pacific Design Center, which is now a Cedars-Sinai Innovation Laboratory, preparing stem cells for experiments and for patient therapy. Not only are we doing our stem cell work in the Pacific Design Center, but you will soon see when the next SpaceX shuttle is launched, that our stem cells from Cedars-Sinai, partnering with NASA, will be sent into space. And those little chips that I showed you with their arterial and venous fluids going in and out and their photography of the molecules will take place in space. And here is a simulation of a technician in space who is loading our stem cells into the next SpaceX launch. And the theory behind this is that the lack of gravity in space is a powerful force on the response of disease cells to certain drugs. And we'll be hearing a lot more about this over the next few years. So another example of the research that's continuing is our heart and stroke programs. And this is probably the slide that whenever I talk about work at Cedars-Sinai that I am most proud of, because this slide really is a most remarkable set of data. This is data provided by the government every year, testing all hospitals in the country, testing mortality after 1 month after acute myocardial infarction or heart attack, or after a stroke. And the curves that you see here, the spread in the blue is the mortality rate in every single hospital in the nation. The national mortality rate for stroke is 13.6% in 1 month. The national mortality rate for acute heart attack is 12.9% after 1 month. Cedars-Sinai is the lowest mortality for heart attack or stroke out of any hospital in the country. This is data which we were sent from the government, which is a reflection of 2018 through 2019. And I can tell you that 2020 data was identical. And I think this is probably the most important slide that I'm going to show you tonight, that if, God forbid, you're going to have a stroke or an acute heart attack, Cedars-Sinai is the institution which could offer you the lowest mortality rate out of any hospital in the nation. Our heart program really is superb, and as you know, one of the top in the world. We have the largest heart transplant program literally in the world. We also have the largest aortic valve replacement program in the world. And our aortic valve replacement, and this is one example published in the New England Journal this year, showing a 5-year outcome of replacing an aortic valve without resorting to surgery, doing it through a catheter. And we're now expanded this program to mitral valves as well. And this really is becoming the mainstay of valvular repair and replacement in the world, with Cedars-Sinai as the pioneer. Another area in heart disease where we really are at the cutting edge is a very scary area for all of us, and that is the problem of sudden death. And we've now developed a database, and we've combined that database with genetic makeup of who is prone to sudden death. And now, because we have the genetic collection of patients' DNA and their families after sudden death, we're able to construct a profile where we can actually predict from the family's DNA which genes are markers of potential sudden death, and offer these patients very, very early intervention with pacemakers before they even have symptoms to prevent the onset of sudden death. So these are two examples of work that's ongoing right now, despite COVID. And then an example of what we're doing in cancer, which again is not science fiction. Over the next 5 years, we are going to be studying 5,000 patients in our innovation center, creating for these patients with cancer what we're calling the molecular twin. And what the molecular twin is we're taking a patient with cancer, we're taking his or her clinical data from our electronic medical record, their imaging data, biological samples from the tumor, including biopsy, including blood, and again, including stool bacterial profiling. We're doing molecular profiling of the tumor, including DNA and protein and RNA. We're creating stem cells from the tumor and testing it organ on a chip and injecting it into mice. We're taking all this data, putting it into a data warehouse, sharing it with external databases from similar patients around the world, and after an integrated analysis, offering a complete molecular twin, an avatar of the patient, herself or himself, and giving the physician a comprehensive report of this entire molecular twin of the patient's disease, so the physician will be able to offer the patient a personalized treatment based upon the entire molecular profiling of the diseased patient. This is not science fiction, and the first patients will be offered the opportunity to join this clinical trial this year as we speak. However, COVID is with us. And we thought that COVID was going away. This is data from Los Angeles County as of yesterday afternoon. You can see that there was a peak of COVID after 150 days, just before the summer, and then it went away, and we were all very, very complacent. And then look what happened. After Thanksgiving, this huge surge. The blue is California, the gold is LA County, and the blue at the bottom, the paler blue, is the city of Los Angeles. Huge surges of COVID after Thanksgiving, and now, right now, tonight, we're experiencing another surge after Christmas. It is social distancing. It is the socializing of families that is causing this, and we are right now in a historic situation never before seen in medical care in the modern era. In fact, in any era. All the hospitals in Los Angeles County are overwhelmed. We have already canceled all elective surgeries, and we're looking at putting patient beds in the most unimaginable places in a hospital, and you've all been reading about it. So we hope that this is only going to improve after New Year, but we're bracing for 4 weeks of a further surge. So this is certainly going to impact on all the good things that we're doing for our regular diseases that afflict all of us. However, RNA has come to the rescue. And you've all been reading about the RNA vaccine. And let me just give you some idea. We heard a beautiful set of talks from this Beth Jacob panel 10 days ago about the vaccine. So I'm not going to spend too much time on the vaccine, but just to tell you that the RNA depicted here in this cartoon in this blue-green coil, all it is a messenger, as its name is, a messenger RNA. It is a messenger which tells the cell to manufacture a new protein. And this new protein in the red is the protein which is one of the fragments of the COVID virus. Our body sees this red foreign protein, creates antibodies, and protects us from further infection with the virus which contains this red protein. And none of us expected the results to be so dramatic. And this is data taken straight out of the FDA data which Pfizer had submitted for approval of the first vaccine. And you can see over here in the blue, after the first injection going out for 112 days, and this has subsequently been extended, after 112 days in the blue compared to placebo in the red, you can see that there is no further infection in any of the placebo patients after the initial few were infected within about 10 days of injection, whereas the placebo receiving patients continue to proceed to develop COVID in the community. And for the physicians in the audience, for those of you who are involved in clinical trials, a result like this is hardly ever seen for any drug. To give you an example, the flu vaccine is only effective in about 50% of patients at best. And here we're talking about a 95% efficacy after the first injection, which is remarkable, almost never been seen before in medical science. But the most remarkable attribute of the Pfizer vaccine, and I believe the Moderna as well, based on the data that we've seen and that's published now, is that they're safe. And this is extremely reassuring for society that at least we have something to the rescue. And over the next few weeks, everybody will be offered the opportunity for the vaccine. Right now, LA County has some strict guidelines as to who should be receiving it in a tiered fashion, but ultimately everybody will receive it, going down by age, and we're looking forward to providing the community with full coverage over the ensuing weeks. And I'm going to end off with a reminder for the old people like myself in the audience. For those of us who were in college and medical school in the 1960s and 1970s, we learned about the flu of the early 1920s, late 1919. And we learned about it as a historic event. And this is a beautiful painting by Morgan Weisling, painted in 2013, before COVID, when he was imagining in a most poignant way what the flu was like in 1920. And you can see this poor little boy, the terror on his face, how he's clutching the blankets. You can see the mother who's really, really concerned and worried. She has the highest anxiety on her face because she knows the ultimate result of the flu in 1920 for a little boy. The doctor, although he can't offer much, and here he's offering a spray for the bronchitis, the doctor has got the solid look about him, offering a really solid and yet not too optimistic and not too pessimistic look on his face. So in 1920, I think Morgan Weisling captured what our parents and grandparents were experiencing. And only 100 years later, we are not facing the same situation. We are facing an opportunity of really preventing and really killing the scourge so that hopefully none of our mothers and none of our children will have the same anguish that this doctor has to deal with in 1920. So I can see that I've spoken for 40 minutes now, and I promised the rabbi I'd give you at least 20 minutes for discussion. So I'm going to stop over there and offer it to you to continue.
So let's indeed open it up for questions. You're welcome to send in the questions through the chat. Also, thank you, Shlomo, for also addressing the situation with the COVID, that we have to maintain our vigilance in such a scary situation, but also encouraging with the development of these vaccines. So we have a couple of questions that came through the chat.
I see them.
And you're welcome, Shlomo, to address both in terms of the vaccinations shortly after birth with babies in Israel given vaccinations shortly after birth, and then Ron Cohen's question about the utilization of the vaccine for other diseases. I don't know anything relating to it, and I'm a layman, but please address. I don't think it's used to replicate the genetic code of the virus. Can you address that piece as well?
Yeah. So the first question about BCG. So yes, I got BCG. As a medical student in South Africa, I got BCG. A lot of Australians did, a lot of South Africans did. In Israel in the 1960s and 1950s, especially when the Yemenites and the Iraqi Jews came to Israel, there was a lot of TB, so they gave them BCG. if you look at the population statistics, that is why one of the reasons that in Africa, although the sanitary conditions and the living conditions are so poor, COVID really hasn't had as high impact as it has had in Western Europe and America. The theory is, one of the theories is it could be anti-TB treatment. It's not proven, it's only epidemiological. In Holland, there's a big study now that those who receive BCG have a lower incidence of COVID. And we are doing the same study over here now. I don't know, but there's no direct evidence, but it's all based on population epidemiology. And so perhaps the evidence would suggest that if you've had BCG, the risk of developing COVID is lower. I'm not saying it's a treatment or a preventive vaccine. Then the question about the RNA. So RNA, and this is one of the myths that's out there on the internet. RNA does not persist in your body. RNA disappears in minutes. So there's no circulating RNA to give you any long-term effects. As soon as it gets into the cell, it does its business, the cell makes the protein, and the RNA is gone. And unlike DNA, for the biochemists amongst you, DNA lasts for millions of years. So fossils are dated by DNA because the DNA of fossils is intact. Whereas if you take RNA and you put it on the table, it'll be gone in less than a minute. So it's a very unstable compound. So there's no danger of RNA continuing to circulate or causing any toxic effects in our body. I hope that answers the question. And then the question about how will we know when we get the vaccine? The county's issuing guidelines. We're now in the tier 1A. 1B will be a certain tier of age group and disease, and then go down to 1C and then 2. We're hoping that the county will allow us to move into 1B very soon, because 1A seems to be almost completed. So I don't want to give a timeline. You've got to ask the county that question. Remember, all the vaccines that we get are not ours. We don't own the vaccine, and we don't decide. All the vaccines are distributed by the county, and the county health department is the one that controls them. And parenthetically, I would be very cautious about any uncontrolled vaccine administration. And please to stick to the county guidelines, both for registration, for follow-up, and to make sure you're getting the authentic vaccine.
Shlomo, there was a couple of questions here. I'm not sure if you just addressed a bit in terms of why aren't vaccinations proceeding 24/7. Also, is the vaccine safe for pregnant women?
Yes. Well, we don't know about we don't know, because the trials did not include pregnant women. So the answer is we just don't know. We don't know about pregnant women. We don't know about babies. Those trials are ongoing right now. We don't know the answer.
What about women before getting pregnant?
No problem. Well, yeah, there's no negative reproductive impact. Remember, this is safer than many of the vaccines which we've all received for measles and mumps and all those horrible diseases which we used to talk about. Because when we got those vaccines, even the polio vaccine, those were dead viruses. Those were real viruses which we got injected into ourselves. This is not a virus. This is an RNA molecule which disappears. And another myth is it's not new. RNA viruses have been around for several years and have been used experimentally for Ebola and for other tropical diseases for at least 3 years in trials. So it's not as if this is a new model. And I cannot overemphasize that the risks of the vaccine are minuscule compared to the risks of COVID. So it's a value judgment. And if you look carefully at the published data, go on the FDA site and see the published data on the vaccine side effects. Look at the placebo column. The side effects in the placebo were sometimes worse than in the vaccine in terms of fatigue and fever and muscle ache. So overall, the value judgment is unquestionably in favor of the vaccine, and I'm glad that that our rabbinic leadership have come out so strongly in favor of the vaccine.
Thank you, Shlomo. Thank you, Shlomo, and it's so important for us to hear your medical expert opinion on that the vaccine is safe, it's effective.
It's not my opinion. It's the data.
The risks.
Rabbi, it's not my opinion. We rely on the data. That's what the data is telling us.
Shlomo, there's another question that came in here in the chat box about comparing COVID to the flu, where there's like different strains of the flu each year. So shouldn't we be concerned that there's going to be different strains of COVID? There's already a different strain that we're noticing, and how is the expectation that the vaccine will be effective against the different strains that might arise?
That's an excellent question. For influenza, not for the other viral, not for mumps or measles or whooping cough, but for influenza, you do need a new vaccine every year. And what the CDC does is they profile the influenza that they're expecting for the subsequent winter. And based on that profile, they design a vaccine for that population of influenza viruses based on what they expect to be the prevalent influenza for the season. So every year we need a new influenza vaccine. We don't know whether we will need annual vaccines for COVID. We just don't know. What we do know is that the virus is mutating. It will mutate. All viruses mutate. And so far, even the so-called virulent mutant appears to be neutralized by the vaccine. It's certainly more virulent. There's no evidence that the patients are more sick. And it does not appear to be prevalent in Los Angeles. I understand that there were 1 or 2 cases of the British variant in San Diego. I don't think there have been any in Los Angeles. There may be, but I'm not aware of it.
It's amazing to hear about this vaccine. I mean, it's miraculous that it's been able to be developed, and you've described how much of an incredible accomplishment it is for the medical scientific world to discover it. And I know this sounds rabbinic, but I'm a rabbi, which is that grateful to Hashem to grant such wisdom to our physicians to be able to discover these vaccines.
You're absolutely right. It's a remarkable feat in a matter of weeks to get it going. However, with all due respect, and these guys are brilliant, with all due respect, they've been working on the RNA vaccine for several years. So they were ready. Luck favors the prepared mind, and they were ready. And they just jumped right in, and kol hakavod to them, and they were able to pull it off. Both Moderna and Pfizer and the others.
Will vaccines for other diseases be replaced with ones based on mRNA?
Yes.
And also maybe confirm for us, the question came in, do we need 2 doses or does one suffice? I think from what I've read, you need 2, but can you speak to that? Do we need 2 doses or does one suffice?
So the honest answer is we don't know. We only know that 2 works. And that's the correct answer. The correct answer is that 2 definitely work. We don't know whether one will work or not in the long term. So that's why the CDC recommendation is that we stick to the 2. By the way, that's the reason why the county is so rigid in their control system, because they want to make absolutely sure that whoever gets a vaccine will have the second vaccine, and the whole thing is pre-programmed. So when you get your first vaccine through the county registration, in hospital setting, automatically you will be slotted in for the second vaccine, and that vial is reserved for you. And so it may be that we don't need the second vaccine, but right now we rely on the data, and the trials were only done with the 2. So therefore we stick to what the trial results were.
Thank you.
Going forward, that may change.
What can you tell us about the other vaccines like Johnson's and Johnson's? And also question here about what about vitamin D and C? Any tests done on that?
I don't know about the Johnson & Johnson vaccine. Vitamin D, I know there's some work on vitamin D. I'm not aware of anything really compelling. Vitamin D has been used in a lot of situations. It was even used for breast cancer once. So I don't know. I can't answer that question. I simply don't know. Now, I have a question here about why are patients dying in hospital right now? Well, remember, those patients wouldn't benefit from a vaccine. The vaccine has to be given at least 2 weeks, before you get the infection. So once you're in hospital, it's not going to make any difference to how you're going to do. Why are they dying? That's a very good question. Their patients are dying of very severe lung damage. Patients are dying of stroke, heart disease, and kidney failure. And the stroke appears to be a very, very severe form of a clotting mechanism which goes awry. And there is an as yet unknown mechanism of the virus to make the blood very viscous and very thick. And all those factors that I mentioned are more prevalent in patients who are obese and patients with diabetes and high blood pressure, and patients who are Hispanic and Black. That's populations that I gave. So that's what we know. We know a lot of questions and very few answers.
Okay, fantastic. Thank you, Shlomo.
I got one question about a VIP patient. How is XXX doing at Cedars-Sinai? I'm not going to answer that question because I don't even know that XXX is in Cedars-Sinai, except for reading CNN. It may not be true.
And Shlomo, you don't need to answer that one. And this was fantastic, and you were amazing. And we just thank you for all the great work. It's such important, life-saving work that you're doing. And may HaKadosh Baruch Hu bless you with good health and wisdom and only good things to continue all the work that you're doing. I'll let you, if you want to add one last comment, Shlomo, you're welcome to, but otherwise just fantastic crowd tonight, both in terms of the amount of people and just a great chevra. We had close to 100 devices and maybe 130, 140 people. So really well attended and so interesting, and such important information that you shared, Shlomo, tonight. So did you want to, you're welcome to just say goodnight or share a last word.
Well, my only plea, because I mean, this is Beth Jacob, this is our family, is please don't rely on all the kooks on the internet. Ask your doctor. Ask your doctor. Your doctor won't mind being called, even if it's after hours. Call your doctor, email your doctor if you have any questions. Please, just, 90% of what the stuff that's out there is not accurate.
That's the first blessing of the Bakasha, of the middle part of Shemona Esrei, is Ata chonen la'adam das, right?
That's right.
We ask Hashem to give us wisdom. And so we also should always seek knowledge and wisdom and, make sure that we're looking after the right sources for that knowledge. So
but we also need the next bracha. We need Rafa'enu.
Amen. Amen. We should wish everyone just good health, stay safe and well. Only good things, and see you again soon. All the very best. Thank you so much, Dr. Shlomo. Fantastic.
Thank you.